I'm Ariana Przensky, and today we're going to show you how to cryopreserve adherent cells so that you can store them for later use. Make sure you clean your workspace with ethanol before you begin.
Now we're going to prepare the cryomedia. Cryomedia is 90% media and 10% DMSO. You'll need to sterilize the cryomedia before using it, so take the syringe filter and screw it on the syringe. Add the media and DMSO into the syringe. To make it simple, we're using 9 milliliters of media and 1 milliliter of DMSO. Once the two have been combined, filter into a larger storage bottle. Make sure you do this quickly but carefully because the DMSO will eat through the filter.
So you need to take your cells out of the CO2 incubator to assess them under the inverted microscope. After assessing your cells with an inverted microscope, discard the old media. Next, you'll have to trypsinize the cells by adding an appropriate amount of trypsin in order to detach the cells from the flask. Make sure you look at the cells under a microscope to ensure that the cells are still attached. Don't leave the trypsin on too long, otherwise you will discard your cells when you're getting rid of the trypsin. Then remove the trypsin and leave about 2 to 3 drops in the flask and assess the cells under the microscope again to ensure that the cells are still there. You'll begin to notice that the cells will round up upon being trypsinized. Once the cells are suspended, deactivate the trypsin by adding enough media into the flask. Then you'll need to complete a cell count to meet the appropriate seating density of the flasks you're using in the lab.
Next, you need to transfer the cells and media into a centrifuge tube for centrifugation. This is done to isolate the cells. Remember to balance your centrifuge with another centrifuge tube that has the same amount of volume of liquid. Once a pellet is obtained, discard the supernate. Then resuspend the cells using 1 milliliter of cryomedia. Transfer the cells and the cryomedia mix into the appropriate number of cryovials. This will depend on your cell count. Be sure to label the cryovials appropriately.
Transfer the vials into Mr. Frosty, which contains isopropyl that helps to freeze the cells slowly. This avoids crystal formation and cell death. After about 24 hours, take the vials out of Mr. Frosty and place them into the liquid nitrogen tank. While handling the liquid nitrogen tank, be sure to wear protective equipment such as your lab coat, goggles, and a thick glove to prevent burning your hand. Remove the stack of containers slowly to prevent spilling the liquid nitrogen. Be sure to keep track of where you store the vials so that they can be retrieved for future use.
And that's how you cryopreserve cells for future use.
Thanks for watching!